So welcome back to the Classics of Immunology Journal Club. Today is a milestone red letter day because we have finally finished with the history of immunology, and we're now in real time. And I'm going to review a paper today that just came out in the Journal of Experimental Medicine. And the authors of this paper are Joey Lim, Anthony Tan, Nina Lebert, Shou Han, Jenny Liu, and Antonio Bertoletti. And this comes from a new institute sponsored in part by Duke University in Singapore. And the title of this paper is SARS-CoV-2 Breakthrough Infection in Vaccinees Induces Virus-Specific Nasal-Resident CD8 and CD4 Positive T cells of broad specificity. And this came out in the JAM in October of 2022. And the background on this paper, and this is what I was going to talk about off the top of my head. Now I don't have to, because now I can just review this paper. But the background is that vaccines, the history of vaccines is important to understand, particularly when we're having such a pandemic and vaccines are so important and so forth. Vaccines, you have to realize that vaccines were traditionally been tested to prevent disease, not to prevent infection. So when we had these new mRNA vaccines from Moderna and Pfizer BioNTech, this was a huge new thing in the whole vaccine world. And you have to remember that there were no tests for the microbes until, just recently. The HIV pandemic was ushered in new molecular biology techniques that allowed the molecular virologists to devise new tests to test for the virus. But before, and in going back to Jenner's smallpox vaccine in 1798, what evolved in the 19th century after 1798 was that they realized they injected the vaccine into the skin, for the most part intradermally. And then what would result would be a delay type hypersensitivity reaction at the site of injection that would run its course over about a week or two and ultimately scar over and would leave a scar in the skin. And one of the ways that people then would test to see whether you've been vaccinated, you had to roll up your sleeve and show them your smallpox scar. But the point is that and this is what Jenner did. He used cowpox to immunize people against smallpox, cowpox being a similar but different virus from the same virus family, a pox virus. And it would set up a local infection in the skin that then was considered to be, when they started to see the inflammatory reaction, the DTH reaction, they said, well, that's a take. And so that's interesting from the standpoint that from the very beginning, Jenner, by setting up a local real infection, all of the mechanisms of immunity and immunology came into play. And because it was a cell-mediated immune response, that's what delayed type hypersensitivity is. So antibodies hadn't been discovered yet and wouldn't be discovered for almost 100 years in 1890 by von Behring and Kitasato. And so from the beginning, T cells were very important in this whole process. Later then, in the 20th century, after antibodies and antibody tests came into being, had been created and developed, they were able to test blood for the presence of antibodies if they had some sort of an assay for it. And one of the very first kinds of assays that was devised was a neutralization assay. And that required culturing cells in the laboratory and adding virus at different dilutions, and then adding virus at one concentration, and then adding in serum diluted out to see whether or not the serum would prevent the infection. And usually it was a cytopathic effect. The virus killed the cells, and that's what they measured. And therefore, neutralization assays came into widespread use. And then clinical studies were shown that maybe that if you had a certain tighter concentration of neutralizing antibodies, that would correlate with protection from disease. So it was protection from disease all along in the 20th century. And not until now have we had really good vaccines that prevent infection. Everybody who's only experienced the COVID pandemic, this is a whole new world. The thing is, one of the things that evolved from this history is that from the time of Jenner, vaccines were introduced into the body through an injection. And in the smallpox infection, that's where the infection became manifest, was in the skin, because you get all these poxes, which were foci of infection and inflammation in the skin. So they naturally injected the vaccine into the skin. And then from there on after, laboratory animals as well as humans and so forth, it was vaccines were always administered as an injection into the skin. However, and so it was a parenteral vaccination from the beginning. And in the 50s, when virologists had developed assays for the virus, cell culture assays, they took Pasteur's advice from 100 years previously, because Pasteur erroneously but correctly told everybody that what you had to have was an attenuated live microbe. You couldn't kill the microbe because then it wouldn't work. And Pasteur's theory was about why that wouldn't work was that he thought that what was going on, just like in microbiology, if you don't have the proper culture conditions for your bug, and if you leave out trace elements or other kinds of things, then you can't get them to grow very well. So what Pasteur thought was going on with vaccines was that if you introduce the vaccine into the body, the microbe would begin to replicate, and it would then consume vital trace nutrients from the body, leaving the body depleted of these trace nutrients, so that when the person would come into contact with the microbe out there in the environment, the bug couldn't grow. And of course, that's not how immunity works. Now we know, as a consequence of von Behring and Kitasato, that the body makes antibodies that deal with the virus in different kinds of ways. And we also know that T cells get activated and they secrete cytokines, and cytokines cause inflammation, and the inflammation then brings the macrophages into the battle. And ultimately, if all things go well, the microbe is subdued. The thing about the skin is that if the portal of entry is in the skin, then it makes sense to vaccinate in the skin or around where the portal of entry is. But in a respiratory virus such as COVID-2, as well as measles, as well as smallpox, the portal of entry is initially in the nasal mucosa, and then in the whole upper airway. And then with COVID-2, it gets down into the lower airway, into the lungs. And once it gets into the lungs that are so highly vascular, it can go every place, essentially, very rapidly. So the portal of entry is very, very important in vaccines. And it's only now, I think, being recognized as to how important it really is. And just before the COVID pandemic happened, there was a paper in the JEM, another paper from the Vaccine Research Center at the NIH. Bob Cedars' group had been working on TB vaccines, and TB vaccines had been, the BCG and so forth, had been notoriously, not very effective. At least the clinical studies never really showed broad activity and specificity and so forth. But what Bob Cedars' group did was they, instead of injecting the vaccine under the skin or into the skin and so forth, they injected it intravenously, BCG. And when you put it in a vein, the vein goes up, dumps it in the inferior vena cava, goes to the right heart. And where does the right heart pump it? Right to the lungs. And they showed tremendous efficacy for then inhaled TB through the nose and the mouth into the lungs, because in the lungs then now they could show that there were all kinds of T cells there that were very good in terms of the rejecting and reacting and rejecting this bug. So that's just as a background in this whole business. When COVID came along and the messenger RNA vaccines were had been developed over the previous 10 years, but they hadn't gone anywhere because the people, the companies, what I call the filthy capitalists, couldn't figure out how they were going to make a lot of money on this stuff until there was a real need for it. And so the mRNA vaccines came into being. And the molecular virology, the molecular biology of this whole thing is that because of the fact now they knew what the cellular receptor for the virus was, number one. They knew that that cellular receptor bound to the spike protein on the surface of the virus, and that there was a receptor binding domain in the spike protein that bound to the receptor on the surface of the cells. And so therefore they used, to make their job easier and quicker, they used a selected portion of, for the most part, of the spike protein, which made for a very specific vaccine. And that if you got good antibodies, neutralizing antibodies, those antibodies would glom onto the virus and prevent its attachment to the cells, and that would prevent infection. And it worked. And so we've, in a short period of time, like a year, a little bit over a year, we started distributing vaccines and other places around the world to combat this virus. And so you all know the story of the pandemic at this stage of the game. But I said early on to myself, and anybody else who would listen, I said, well, what about if you make it so specific, you're going to get escape mutants. That virus is going to replicate, it's replicating really rapidly. It's Darwinian selection for resistant mutants, so that don't bind those antibodies, but still bind the cells, and the virus can just go on its happy way. Well, that's exactly what happened with the different so-called variants. I don't know why they haven't called them mutants, so that the lay population realizes that we have genes that get mutated, and that without that we wouldn't be here. We'd still be back in the slime mold, because evolution depends on mutation. It took about a year or so, and that's what was surprising to me. I thought, this isn't going to work all that well, because we're going to select out for resistant mutants right away. Well, they did, but it took a while for the mutants to be selected for. But once they were selected, they ripped through, the UK and Europe and South Africa and so forth, and the United States, until, in a very rapid fashion, those resistant mutants became the predominant strains of virus that were replicating in the community. This paper that was just published, in the introduction of this paper, these people point out several things that are really relevant and cogent for this whole thing. And they said, number one, the upper respiratory tract is the portal of entry, and the nasal ciliated epithelial cells in the nose and in the nasopharynx have large amounts of the receptor for the virus. That's number one. It's a setup for the virus. The other thing about this virus that has not been really widely publicized, that I said early on, because smallpox vaccine and measles virus, or smallpox virus and measles virus, they both have genes that disarm the innate immune response. And what is the innate immune response? Well, primarily it's from virus infected. When the virus gets in and infects cells, it activates the production of interferon, alpha beta. And the viruses, because it's a quest for survival, to survive the immune response, the viruses have generated and developed and picked up genes that will inactivate the interferon alpha response. They basically, a lot of them block production of interferon. So early on, after infection of a human being or an experimental animal, what happens is that the virus inhibits the initial immune reaction, and it just replicates, like home free. Come on, y'all come. This is a great host that we've got now. We can just replicate to our heart's content. And so that's why there is a week to 10 days to even 2 weeks of the so-called incubation period or prodromal period before there's any symptoms, because the symptoms are due to the host inflammatory response to the bug, not to the bug per se. And that's another aspect of COVID-2 and COVID disease that I think most practicing physicians still haven't really come to grips with. The symptoms are not due to the presence of the bug. The symptoms are due to the host reaction to the presence of the bug. It's very, very important because it disarms that interferon alpha response. The only thing you got to hope for after that is T cells. And so not until the T cells come into the fray after a week to 10 days to 2 weeks, because they have to be antigen selected, and then they have to proliferate in response to that. So you build up your army. And then when you do that, by the time that all happens, the virus is replicated to very high copy number. And now you have a huge battle going on between a high concentration of virus and lots of T cells. But the good thing about the T cells is they also make interferon, but it's a little bit different. We call it interferon gamma. And the interferon gamma stops eventually the replication of the virus, the widespread spreading of the virus and so forth. And the T cells have other little attributes like the ability to kill infected cells. And so the T cells go about their business in a sort of like a hand-to-hand combat. They're the Marines with the bayonets fixed, and what they do is they go along and they kill off virus-infected T cells, infected nasal epithelial cells and lung cells as fast as they possibly can. And so that's how that whole process works. And so the thing is that resident T cells, if you survive an infection or if you've been vaccinated, you should be left with resident T cells in the tissues, particularly at the portal of entry, if the vaccine has been given to the portal of entry. But of course, we don't do that. We're still back in 1798 injecting virus into the skin. Vaccine, I'm sorry. The other thing that happens is that there's a fair amount of data on the inflammatory cells that come into the lung in response to COVID, because they can do bronchoalveolar lavage and wash out the lungs, the bronchi, with lots of fluid, and then they can harvest a fair number of cells from the bronchi. But the nasal epithelium up in the nasopharynx is a different story, and there haven't been too many studies that have been done on the nasal epithelium or on the nasal passages and so forth until this study. And this study, I feel, is perfect for the Classics of Immunology Journal Club, because I think this paper's a classic. You can read it and then disagree or agree with me. So let's go to screen sharing. That's the introduction of the paper. And so this is figure 1 from this paper. And in the A portion, and these panels up here, it basically shows them how they did their experiments. They did nasal swabs that they put into tissue culture medium, and then they could wash the cells that they got out of the nasal passages and so forth. And then they could test them with a flow cytometer, and they tested the T cells, CD3, CD4, and CD8 monoclonal antibodies. And they had a couple of antibodies, CD45RA and CCR7, which they felt that identified memory cells. And then tissue resident markers, CD103 and CD69. And so that's what they did. And in order to go beyond just phenotyping cells and actually test their functionality, here is the bar depiction of the genes of the virus. And so they have an open reading frame here, a non-structural protein 12 here, and then structural proteins here. And in red is the spike protein, and then they're blown up here. The structural proteins include membrane proteins and a nuclear protein. And so they selected out then the membrane, the nuclear protein, the spike, and a non-structural protein, number 12, to test. And they made peptides from these genes and then used those peptides in a T cell activation situation whereby they had T cells in wells and they would pulse them for 3 hours with their peptides. And the positive control was anti-CD3 and 28. And the way they tested these were by ELISpot, looking for interferon gamma for the most part. They also looked for IL-2 and secreted into the medium. So what they found then, just in the phenotypic characterization, in peripheral blood mononuclear cells, they looked for what they thought were their tissue resident markers, CD69 and 103, which are also activation markers. PBMCs were essentially negative, whereas the nasal cells from the nasal T cells that they were able to get out, they were all positive for these resident markers. And then if they looked at CD4 versus CD8 in the PBMCs, they have CD4 is green and CD8 is orange. And so most of the peripheral blood cells are, it's usually a 2-to-1 ratio. And here we are 60% and 30% essentially. Whereas it was reverse in the tissues. Most of the cells were CD8 positive cells and not CD4 positive cells. And then here's they looked at different patients and so forth and compiled the data. And here's CD103 versus CD69, and you can see in the nasal CD4 positive cells, 78% were double positives, and in the CD8 positive cells, 97% were double positives. So then if you go to the second figure, when they looked at individuals that were naive, but vaccinated against solely the spike protein, if they looked for cells that would secrete interferon gamma in response to the viral peptides, in the nasal mucosal cells, they had 16 of those, and then they had what they call convalescent, and those were these people had at least 2, but mostly 3 immunizations with the mRNA vaccines, and then they had convalescent people that had been vaccinated, but then had breakthrough infections, and they had 22 of these individuals. And so basically, these are the vaccinated people, but not infected, and these are the vaccinated and infected people. And you can see that the interferon gamma positive ELISpots were non-existent for the nasal cells, were non-existent in the nasal population. So that even though these people were protected from infection, they had no cells in the nasal mucosa that could recognize any of these peptides, including the spike in the nasal mucosa. And by comparison, the patients who had had breakthrough infections, so that they had been exposed to all of the proteins or all of the gene products of the virus, you can see that they had varying amounts of ELISpot positivity in the whole thing. And most of the patients had reaction to at least 2, 3, or 4 different gene products. And here you can see these are pictures of the ELISpot. This is a positive control. This is the naive vaccinated people. It's all negative. And here is the nuclear protein, the spike protein, the non-structural protein. And here's the positive control. The next figure shows I like this figure, the difference between the nasal cells, T cells from these people versus what's going on in the PBMC. And of course, they're testing different genes, the membrane, the nuclear peptides from the genes, the nuclear protein, the spike, and the non-structural protein 12. And these are nasal cells, and these are all people who had breakthrough infections. And these are their peripheral blood cells. So you can see that the nasal cells that are all activated resident T cells are reacting to many of these different peptides, whereas for the most part, there are very few reactive cells, even to the spike proteins in these individuals in the peripheral blood. And so for the most part in immunology today, and when you're dealing with humans, you only study the peripheral blood because it's difficult to get enough cells to do anything with from other parts of the body. And here in B down here, we see the LE spots in the nasal cells, in the NSP12 here is very positive. So that was figure 3. Now in figure 4, they studied how long these cells remained positive in response to their peptides. And they found the months post-recovery in breakthrough infected vaccinated people, they stayed positive for at least 3 months, but they started to fall off after 3 months towards 6 months. And here's 3 different people that were studied on multiple days afterwards. And you can see, these are, the red is the spike protein, and then these are different structural other proteins. And you can see that they differ, but they remain positive for at least 90 days after the infection. That's it. And now I'll stop the screen sharing. And in the discussion of this paper, they make several points. And they list them out. And then they say the breakthrough infections of COVID in these vaccinated people leads to the detection of nasal T cells that are specific for many of the peptide gene products of this virus. And that's number 1. Number 2 is, in contrast, using the identical assays in the nasal T cells in vaccinated but uninfected, no breakthrough infections. There are no antigen-specific T cells, including the spike protein. And so the idea is, and this is what they speculate on in their discussion, is that multi-antigen specific T cells might rapidly trigger interferon gamma secretion, which would then function to decrease viral replication at the port of entry into the nasopharynx. And they call this hybrid immunity then, because you have to be infected in addition to getting the vaccine. And they figure that that's the reason that if you get infected with the Omicron variant and the vaccines didn't cover the Omicron spike protein, that you can recover from the infection much more rapidly, because you got all these T cells there that are making interferon gamma, dampening down the replication. The bottom line, that my conclusion of this is, we need to construct vaccines with multiple genes to stimulate immunity against many peptides of the virus, just like happens in natural infection. What a concept. And we need to have peptides focused on in these vaccines, in these new vaccines that are ultimately going to come. Have to. We need to have them stimulate both humoral immunity, antibodies, and neutralizing antibodies. But in addition, we need to have them stimulating T cell immunity. And of course, the T cells I've always felt that they were better than the B cells, because there are T cells in our body up to 500 billion T cells that can react to all kinds of peptides. And so a natural infection or a vaccination with many different peptides will result in the clonal expansion of multiple clones of T cells that will then be standing there on the ready to make interferon gamma and to prevent the initial replication and to prevent that prodromal period where the patients are asymptomatic. They don't even know they're infected, and the virus is replicating like gangbusters. So I'll stop there. And don't forget, I forgot to mention it at the beginning, don't forget to like the video on the YouTube channel and to click on the bell, get notified for more, subscribe. And because now we're going forward in immunology. We're going to focus on papers that I will pick out and identify that I think are really, really important for all you present students of immunology. So I'll stop there. Thanks for listening. If you're new to immunology, or you just want to delve into a certain aspect of this large field, and you want to get a grasp on things, you should buy my new book, Molecular Immunity, because it's a chronology of all of the important experiments and discoveries that have been made in the last 60 years. It's concise, but it's also very comprehensive. And it's the only such book that's been written since 1970. So you can buy it on Amazon, and together with the YouTube videos of the Classics in Immunology Journal Club, you can't go wrong.